Hi, this is Miss Litton, and this is 7th period review, APBio, Unit 7 essay. Say hey.
Hey.
That's very cute. All right, so the first part is the kidney plays an important role in maintaining homeostasis in humans. Now, you could lead with some introductory discussions right at that point, talking about how it contributes to homeostasis, right off the bat, just as an introduction. But then, when I break down the essay, I said describe the macroscopic and microscopic structure of the kidney. What would you think is the macroscopic structure? Macro means you don't need a microscope. Yes? Yes, cortex, medulla, pelvis. Also, you would talk about what vessels enter and exit it. So what vessels enter and exit the kidney?
Oh, the renal artery and the renal veins.
Renal artery enters, renal vein exit. What else? Yes? Ureter exits. So you would want to name all the tubes and like what is in them, the structure of them, that you have 2 of them. Okay? That would be the macroscopic structure. The microscopic structure, you're going to talk about what? Yeah. And you're going to talk about the nephron. You're going to talk about where it's located in the kidney. You're going to name each part of the nephron. You're going to start with explaining about the blood flow versus the, oh, and I kind of tell you that in B, so I won't skip ahead. But you would talk about the nephron, you would talk about the structure of the nephron, the glomerulus, proximal, descending, ascending, distal convoluted tubule. You would talk about the collecting duct and that other nephrons are associated with that collecting duct. And that collecting duct ultimately drains into the pelvis, to the ureter, to the bladder, to the urethra. Okay, so you're going to set up the anatomy. Okay, in 1B, explain the process of urine formation as well as the hormonal interactions. So the process of urine formation, remember when we broke it down into 3 parts? What are the 3 parts? Glomerular filtration, tubular reabsorption, tubular secretion. So that would probably be a really good way to break your essay down on the formation of urine, is explain each one of those steps. So glomerular filtration, you're going to talk about the hydrostatic pressure, right, the force in the blood due to the heart at the glomerulus, and that it is a ball of capillaries, and that forces everything out of the blood, good and bad, except for form elements, right? What are the formed elements? Red blood cells, white blood cells, platelets, okay? Everything else is going to go out. And you could mention that if you urinated that, if that was your urine right there, you would die. Primarily because you would lose too much what? Water. So then, now you're giving me an explanation about part 2 of urine formation. What's part 2? Reabsorption, okay? So, peritubular capillaries, which surround the loop of Henle and the proximal and distal convoluted tubules, their job is to recover. And since you cannot actively transport water, then how do you get the water back?
Salts.
Yeah, you got to move salts and water will follow. And so I basically would break down each section of the rest of the nephron and say what happened at each section. So I would start with the proximal convoluted tubule and say something like 67% of the salts are actively transported out of the tube and back into the tissue fluid and blood. And so 67% of the water follows. Who else goes? Glucose. Yeah, you get reabsorption of glucose there as well. Okay, on the descending loop of Henle, the way it is structured, only what comes out? Water. And then you would say the water is coming out at the descending loop of Henle due to at the ascending loop of Henle, water can't come out, but salt is both passively and actively transported out of the ascending loop. Okay? Then I would talk about the distal convoluted tubule, that both salt is pumped out and water follows. And then at the collecting duct, what happens? Oh, no, we're not there yet. The collecting duct, water is reabsorbed and some urea. Okay, so that's taking care of tubular reabsorption. Okay, and you want to be clear that it's getting taken out of the loop of Henle or the rest of the nephron and put back into the blood ultimately. Now, step 3 of urine formation is tubular secretion. Now, tubular secretion is where you actively transport out of that urine anything you want, or sorry, out of the blood that you want to put purposely into the specifically distal convoluted tubule, but it can happen in other ways, other places as well. And then I have a couple of examples of what you would want to get out of your body and put in. That might be a good time for you to also mention how the kidney's role, in besides water balance, which doesn't hurt to overstate, is talk about the pH of the blood, depending on the amount of bicarbonates and hydrogen ions excreted or reabsorbed. And you can make a connection between water balance and blood pressure too, right? Okay. All right, that is number one. For number two, nervous control, describe the following events in some detail. Describe how an impulse is transmitted along a neuron fiber, how it reestablishes the resting potential. Then what happens at the synapse and what a reflex arc is. So if I was going to talk about this, I would probably break it up into describing the resting potential. And so I would identify what the resting potential is and what it's caused by. So I would start with that. So what is the resting potential caused by? Three things. Sodium potassium pump.
Negative ions and proteins.
Yes. And the, okay, so sodium potassium pump, you're going to have to talk about how you're actively talk about what a pump is, using ATP to move a substance against the concentration gradient. That these are small particles, but because they're charged, you have to use a pump, you have to use a protein, and it's going to cost you energy. You're using a protein because it's a charged particle, and it costs you energy because you're going against the gradient. That's why you need a pump. And then it's not equitable. You have 3 sodiums out for every what? 2 potassiums in. So that right there creates a differential. And then what's the second one we talked about? Negatively charged ions and proteins inside, and you can give an example, like chlorine. And then what's the third one?
Leaky potassium.
So you want to say, don't just tell me leaky potassium gates. That is exactly what you would write down, but then follow it up with, because potassium is more concentrated inside, the gates don't seal, some potassium goes back outside, based just straight up on diffusion, right? So it wants to go out there, so that's another thing that makes it inequitable. All right, so that tells me the resting potential. Then I say, how does the impulse travel along? Assume you have an EPSP, an excitatory postsynaptic potential at that point. So what's happening is you have ligand-gated sodium channels opening, sodium's coming in. If it reaches the threshold, then along the length of the neuron, so I would mention threshold, sodium, ligand-gated sodium channels, threshold, and I would talk about that then the voltage-gated channels will open up along the length of the neuron in sequence, and that sodium rushes in. And then describe how a neuron is quickly reestablished. What are you going to tell me?
Potassium going out.
Potassium goes out. And what would be important to mention so that the impulse goes in one direction? Did I hit record? Oh, I did hit record. Okay, good. What? Overcorrection. Yeah, it leads to the refractory period, where it becomes even more negative than the resting potential inside. So that inhibits sodium gates opening up the other direction. And then the long-term fix, you go back into your, sodium potassium pump, negatively charged ions, but that's still in there. That's to get you to the back to the resting potential, but you just got to do the sodium potassium pump there. Okay, the events that occur at the synapse when an action potential arrives. So bring your action potential to the synapse if you get that question. Okay, so talk about an action potential is traveling along the length of the neuron. You're at the axonal bulb. Sodium gates are opening. Now, at the axon, what gates open up? Calcium. And you want to mention that calcium's going, where the calcium is when the calcium's opening, the calcium is concentrated outside the neuron and it's moving into the neuron. And then calcium will fuse with the what? Synaptic vesicles, and you could even specify it's the presynaptic vesicles, right? Synaptic vesicles on that, which then migrate to the presynaptic membrane and release their contents into the cleft. And you would tell me the cleft is a small space between the pre and the postsynaptic neuron, right? So make sure you tell me what the cleft is when you talk about it. That's how you get over the points you need for every essay. Then, what's that neurotransmitter going to do? Bind to receptor on the postsynaptic membrane. Now, I can't remember how I asked it. Okay, so then I would break it down into two parts, part A or part B. What happens? Because it's either going to be an EPSP or a IPSP. So differentiate between those two. Say what they are. For it to be excitatory postsynaptic potential, what has to happen inside the neuron? It has to get less negative, okay? Or the outside has to be less positive, yeah. And so that's what that neurotransmitter will affect. And if it can allow sodium to come in, that could be an excitatory postsynaptic potential. If it reaches a threshold, remember we also said about the trigger zone or the axonal hillock? You know, and so the cell body closest, the part of the cell body that's closest to the long axon, which would actually be the one that fires, the closer you are to that, the more your vote has. And it would be a good idea to mention that it depends on how many synapses you have, right? A single synapse is not going to reach a threshold of a neuron. So show that you know that and just say there would be multiple synapses and there would be integration on the postsynaptic neuron. Use a word like synaptic integration because it's adding up all the EPSPs and IPSPs to decide whether to fire or not. You want to say something how the neurotransmitter is recovered, otherwise it would be perpetually firing that neuron, right? And then IPSP, inhibitory postsynaptic potential, if that's what comes out due to synaptic, you know what, as I'm thinking about it, I would write the synaptic integration and then talk about the two parts, whether it could be EPSP or IPSP. Does that make sense? That would set up the scenario of integration. And then you can talk about an IPSP and talk about that it would make the inside more negative or the outside more positive. So the voltage gated channels don't open up. All right. Are we good on that? Okay, so let's talk about the reflex arc. So I would pick like a really easy example like touching a hot pan. Okay? So you want to, the hot pan would represent the what? Stimulus, okay? And that would trigger what? Sensory receptors in your fingertips, which would, there would be dendrites there, right? Travel along the length of the axon. And remember, we don't have to worry. It either fires or it doesn't fire. It reaches the threshold at the dendrites, okay? And then it would synapse in your spinal cord with two types of neurons. What are the two types of neurons it would synapse with? Interneuron that's going to your central nervous system, and a motor neuron going to your effector. In this case, it would be a muscle. Okay, so you could say that at the same time it's synapsing with both. So your effector, your muscle contracts to pull away from the pan. At the same time that your interneuron's going to your brain, you're somewhere in your parietal lobe and you're sensing that the pan was hot. And you could mention how this would be an adaptation, right? So that your hand is removed. You don't wait for your brain to process some extreme pain, whether it's hot or cold or damage to tissues, you respond right away. Or an object coming at you. Okay? You could add little details in there, like the cell body for the sensory neuron is in the ganglia, the dorsal ganglia, outside of your spinal cord. That'd be a little something you could add in there. And the cell body for the motor neuron is located in the gray matter of the spinal cord. All right, that is 2 A, B, C, D. 7, let's say for class, that is not one where I would ask you to explain. That was small print, right? Vision, hearing was small print. So you wouldn't have to write all of that out. So don't stress out about 7A or B. C, I would. I would pick like 5 distinct structures in your brain and where they're located and what they do. That's what I would do. And pick your top 5. And I'm not, yeah. But Do you remember one time I asked you a question, like, tell me your 3 top organelles, most significant? Some of you gave me really stupid ones, no offense, but you did. Maybe nobody in this class, in this room right here. But I'm like, really? You're not gonna pick nucleus? I think I'd pick nucleus. I think I'd pick mitochondria, you know? So pick some, you know, so be like centrioles. Thinking, no, I wouldn't think those are the top. So pick something that's important in your brain. Get 5 different ones and do those. Okay, 8. Can we move on? Are we good? Okay, 8. Describe the anatomy and physiology of the hypothalamus, the pituitary gland. Name and describe the function of each hormone they secrete. Be sure to include a discussion of negative feedback mechanism. Easiest one on negative feedback. What do you think I'm going to say there?
Oh, like negative feedback. Oh, that's
No,
positive.
birth is positive.
Glucose?
No, negative feedback where you're dealing with the hypothalamus and the pituitary gland.
Dealing with temperature?
No, hypothalamus, pituitary. Thyroid. Remember? Thyroid releasing hormone, thyroid stimulating hormone, increases thyroid hormone, and then it exerts negative feedback on both the hypothalamus and the pituitary. Does that sound familiar to you? Okay, so, kind of. What do I have on here? Nothing. Okay, so here's our hypothalamus, right? Hypothalamus is here, and then we have our stalk, and we have our pituitaries. Okay, so on your hypothalamus, let's see, let's make this posterior pituitary, and let's make this one anterior. Oh, I didn't mean to say that. Blue. Anterior pituitary. Okay, now the hypothalamus is directly connected to which one? Yeah, so it's directly connected to the posterior pituitary. And so, in fact, it makes the 2 hormones that the posterior pituitary both stores and then secretes when triggered by another set of neurons. What are those 2? Oxytocin, ADH. Okay, so those are generated by the hypothalamus's neurosecretory cells. Remember this part? And it extends down into the posterior pituitary. Those hormones are stored into the posterior pituitary, and they will be there. Your book doesn't go really into the detail of it until stimulated by just a regular neuron to have them be released out of the posterior pituitary. So oxytocin, what do we remember about oxytocin? Not cervix. That doesn't contract. Uterus. Yeah. That's okay. Cervix. Yeah. It does push against the cervix. Okay. So uterus contracts. And what else do you have? Release of milk. Okay. So milk is out. What? Did I do something wrong?
No.
Okay. So milk is out, baby's out. And that's easy to remember because the baby's coming out, we better feed it. Right? Okay. So that's a direct connection between the hypothalamus and the posterior pituitary. The anterior pituitary, there is not a direct connection. It only extends as far as what? Yeah, the capillaries in the stalk. And then there's a second set of capillaries here, right? And so what it puts into the stalk right here, such a hideous drawing, I can't even stand it, are releasing hormones. And then when these releasing hormones go away, Arrow. When these releasing hormones get carried by the blood and come out into the anterior pituitary, then the anterior pituitary knows to put stimulating hormones into the blood. And these stimulating hormones go to the rest of the body. Are you okay? So let's talk about what kind of stimulating hormones there are. There are what? Yeah, thyroid stimulating hormone, right, which is going to target the thyroid gland. What else do you have? So you have TSH, ACTH. Remember what that one is? Adrenal corticotropic hormone. It's okay. It stimulates the adrenal cortex. Yes.
Are all of them? Like,
yeah.
Mm-hmm. Okay.
Yeah. What else? There's FSH and LH. Those are gonadotropins, stimulate the gonads. Growth hormone. What does that stimulate? Yeah. In what? Muscles and bones. What else? Prolactin. What does prolactin do?
Doesn't it have something to do with milk?
Yeah, it makes the milk.
Okay.
Yeah. 1, 2, 3, 4, 5, 6. I think you have another one on there. And there's another one that could be listed, MSH, it stands for melanophore stimulating hormone, which has to do with water balance in some organisms, and it also changes the color of like animals that change color on their skin. But I'll put that in parentheses because I don't think that was on the notes.
Yeah.
What?
Yeah.
You have a picture in your notes about this.
But not this one.
Oh, okay. Yeah, because this is a really good picture. Oh my gosh, it's embarrassing that you're taking pictures of this hideous picture.
We love it.
It's still better than
It's the
mine.
next public concept.
Oh yeah.
It's abstract, right?
Yes.
It's real abstract.
It is abstract. It's modern art.
Melanophore. Maybe that's it. Okay. Let me just double check because my brain is leaving me right now.
Abstract always like when you just do a lot of like random lines.
No,
That's
that's not the definition of abstract. Okay.
That's what basically.
Oh, I did do them all. Okay.
Yeah.
That's good. All right. So going back, it's important with the anterior pituitary, okay, that you realize that because this is a neurosecretory cell right here, because it's not directly connected, it only gets as far as the blood, and then it puts in the releasing hormones. Releasing hormones carried by the blood go out into the tissues of the anterior pituitary. Anterior pituitary puts in stimulating hormones, and these are the stimulating hormones that I want you to know. And then these are carried to the rest of the body. So if you have, for instance, let's make a person.
Whoa. Whoa.
Oh my God.
You should be an art teacher.
Look at the arm. Why is one of its arm longer than the others?
The head. Arm is looked over.
Here's your hypothalamus, then hanging below your hypothalamus is your pituitary. What else are you doing? And here in your neck is your thyroid gland. Okay, so hypothalamus secretes thyroid releasing hormone, which then triggers your pituitary to start secreting what? Thyroid stimulating hormone, which then triggers your thyroid gland to start secreting what? Thyroid hormone. And as these levels go up, then they exert negative feedback on both the thyroid releasing hormone and the thyroid stimulating hormone.
And then it's kind of like the calcium, back to like the operon unit, when like calcium kind of leads to the transcription factors that digest itself.
Yes. Sure. And then this, for instance, like this would be a really good essay question for you to see if you could respond to it. Is I had a really close girlfriend who's talking to me about her son just last week, and she said he does not have enough thyroid hormone. So then I would, she actually said he had a hyperthyroidism, like hyper. She said a hormone was too strong. He had one of his thyroid hormones was too strong. So when she said thyroid hormone, I'm immediately thinking thyroid hormone, and I'm thinking that her son is creating too much thyroid hormone, which would be like Graves' disease. Remember we saw the eyes were popping out of the head? Do you remember that part? Did I show you that picture yet in your class?
No.
Maybe not in your class.
We don't
Okay.
need to see this.
So I was thinking Graves' disease because she told me he was hypersecreting a thyroid hormone. Well, as it turns out, she calls me back later and says, wait, his T3, which is a thyroid hormone, was not enough. And I said, so when you said thyroid, you meant thyroid stimulating hormone. He was yes. So that told me then, if they have too much thyroid stimulating hormone, that means thyroid hormone is not exerting any negative feedback. His actual thyroid hormone, he was hyposecreting thyroid hormone and hypersecreting TSH because he wasn't doing anything to shut it off. So he's getting more and more and more thyroid stimulating hormone. What's going to happen to the size of his thyroid gland? It's going to grow. Yeah. And so you have to look to see what is triggering what and why we have it in place. Because if you don't have enough thyroid hormone, it makes sense then that your thyroid stimulating hormone, you would have more of it. And chances are you'd have more thyroid releasing hormone as well, right? Because you're trying to get that thyroid gland to start making some thyroid hormone. Yes?
So he had too much TSH.
Yes. This was what was high.
Yes.
Is stimulating
which
hormone.
Signals the thyroid gland to produce
Thyroid hormone.
So he had too much thyroid hormone in his blood.
No, he didn't have enough. She called it thyroid hormone because she knew the first one. So this is what he doesn't have enough of. He doesn't have enough thyroid hormone.
How?
Well, because his thyroid gland is not producing it.
Oh, okay.
Yeah. Now, he's not lacking iodine. If he was lacking iodine, then he'd have a goiter. Now, it still will enlarge. And so he will have to physically take thyroid hormone via a pill. So it will exert what? Negative feedback on his hypothalamus and his pituitary gland. And that's what birth control pills are. You're taking progesterone and estrogen, which normally you would only have if you were pregnant at those levels. So it exerts negative feedback on your hypothalamus and your pituitary so you don't make another egg ready. It's like a false pregnancy. And this is why you might gain weight when you take it or have other symptoms that you would have like you would if you were actually pregnant, because you're telling your hypothalamus and your pituitary a lie and telling them that you're pregnant by maintaining those high levels. Oh, see how much you learned? What? Go ahead, ask me. Okay. And those are the essay questions that we are prepared to discuss today. Good job, guys. Glad you were here. Yes, go ahead.
There's a couple for development.
Wait, say it again?
There was a couple essay questions for development.
Oh, yes. You know what? I forgot because I modified it. So let's talk about that once we talk about that chapter. I believe I have development on the schedule for Wednesday review. Okay? All right, questions, concerns? Yeah, because it would be preemptive to discuss it now before I talk to you about it. Okay, we're all done. Good job, guys. Have a piece of toast.
Yes.
Oh, I'm so embarrassed by this drawing. I just have to tell you, I'm mortified.